So this is probably 5.1 notes. So the functions of the cell membrane are it's a protective barrier, it transports needed materials in, it transports waste out of the cell, it's structural support for the cell, it signals other cells and proteins, so it's a form of communication. The cell membrane is also called the plasma membrane, and it's made up of a phospholipid bilayer, which is two layers of lipids. So remember, bi meaning two. We see bi, that refers to two. So this two-layered arrangement of phosphate and lipid molecules that form a cell membrane, this membrane of lipids and proteins forms a boundary of the cytoplasm of a cell, and that regulates the passage of molecules in and out of the cytoplasm. So this phospholipid, it's kind of like this, referred to as this circle. It has two parts, which is the circle, also known as the head. This is called the glycerol head, and that's hydrophilic. Philic meaning water loving. So this is made up of this phosphate group. So you can see the phosphorus right there, surrounded by oxygen, that's phosphate, and then glycerol. And then the bottom part, known as the fatty acid tails, this one is bent. And if you remember, this one is straight. So if you remember, straight is saturated fatty acid, and the bent one is the unsaturated fatty acid. So hydrophobic means water fearing. They have a fear of water, so they will repel water. Repel water. The hydrophobic lipid ends, so that is the hydrophobic lipid ends, that's this part right, the tails right here. In blue, the hydrophobic lipid ends will face inward, and the hydrophilic phosphate ends face outward. So here we have a picture of a cell membrane, and we call it semi-permeable, but I just want to address a few things here. Again, here is the glycerol or phosphate head part. And then those are the fatty acid tails, which are hydrophobic. The glycerol is hydrophilic. So the red is hydrophilic and the blue is hydrophobic. And then remember, it's a two-layer. So we have that first layer, and then we have this other layer. There's the hydrophilic end, and here's the hydrophobic end. And so this part is inside cell, or the cytoplasm. And then this side in blue is outside cell, also known as the extracellular fluid. That's outside the cell. So we just mentioned that the cell membrane is semi-permeable, but what does semi-permeable mean? So you can kind of think of it like this colander right here, and you're washing a salad, some lettuce, and it allows some things to go through the colander and some things do not. So basically it allows certain substances to pass through the membrane, but not others. And so what affects the membrane, a couple of different things. It could be the size, it could be the polarity, it could be the electric charge, it could be size, polarity, charge, mass, which also is kind of like size. We also refer to the cell membrane as a fluid mosaic model. So what does that mean? That means it's a flexible layer. So we think of fluid as not being kind of a very structured. It has some flexibility or move to it. It's made of these lipid molecules, and they're interspersed with large protein molecules, and they act as channels through which other molecules can enter and leave the cell. So again, here we have these lipid molecules. I don't know if I have any. Yep, here it is, one glycolipid molecule. And they're interspersed with these protein molecules that are in purple here, and there's some different ones. This is known as the integral protein. This is a peripheral protein. And then we also have a glycoprotein. It also has cholesterol, which is a macromolecule right here. So what are the functions of the parts of the cell membrane? So we have these integral proteins. They're used in transporting material in and out of the cell. The peripheral protein, they help the integral protein do its job. Then we also have cholesterol, which provides structure and flexibility. Glycoproteins, that's for that cell-to-cell recognition in the immune system. Basically, they can kind of identify, it's kind of some communication, but what it does is it identifies the cell as being normal or if it's being foreign. And so sometimes if we get foreign cells in our body will attack it and kill those cells. And that's exactly what happens if you're getting a organ transplant that your body recognizes as being foreign. Because of the glycoproteins, if they don't match up right, it says, okay, this is something that's alien, that shouldn't be in the body, and then they'll attack it and kill it. It also has this bilayer or this outer protective boundary. So again, these glycoproteins, they're proteins within the cell membrane. They have long exterior arms, and they're often attached to carbohydrate chains or sugar glycogen. Again, they're used to communicate within the immune system. They're used to identify if a cell is foreign or not. And it also can tell what cell type it is, if it's a lung, if it's a heart, if it's a skin cell, whatever that is. Okay, so they just give a lot of information that the immune system can recognize. And so we call this cell signaling. And that's kind of an important idea to know, the cell signaling. This is the same reason why you can't just get blood transfusions from everyone unless you have a certain blood type that allows for that. So some people might be a universal donor and they can give their blood to anyone, and someone might be a universal receiver and they can get anyone's blood. But some people can only get certain types of blood, and if they get other blood transfusions, then their body would attack it, and that's because of these glycoproteins that are on the surface membrane of your blood cells. All right, so what I'd like you to do is draw a cell membrane in your notes. And no, it doesn't have to be this kind of three-dimensional. You can simply kind of draw it like, you could have, draw these. Actually, I should use the same colors. I think we said red for the glycerol head and blue for the fatty acid chains. I'll draw a couple here. And then you should draw, it's a bilayer. And then we should draw some integral proteins. And then also some peripheral proteins, which help the integral. Maybe draw some cholesterol. we should also draw some glycoproteins. By the way, this is inside the cell. Or the cytoplasm. And this is outside cell. Or the extracellular fluid. And then on the glycoproteins, there's usually some sort of chain, carbohydrate chain, and those are kind of the markers. So your next step here would be to label everything. And I'm going to go refer back to the picture for that, for your labeling. So make sure you label on this the glycoprotein, the carbohydrate chains, extracellular fluid. Oh, we forgot to add this glycolipid. You should put that in. And this cholesterol, peripheral protein, integral protein. You can also add, this is the cytoplasm. Let me see what other things I missed. You could put cytoskeleton if you want on there. Those are used for structure. And so because this is a fluid kind of movement, we do need some structure so it doesn't just collapse on itself or also explode out. So that's what those filaments of the cytoskeleton that helps give the cell its structure. And when you're done with that, when you're done with your drawing, hopefully it looks better than mine. This is where we're going to stop.